And welcome. Welcome. Let me turn off Eye the Mountain before they start singing the next song. Oh, funky bass. Here we go. So it's Monday. It's not sunny. It's even a little snowy. I think I've managed to fix my collar this week. And today we are going to be talking about some delicious things and some not so delicious things. We're going to talk about, we're staying within the arthropods, and we're going to be talking about crustaceans, and we're going to be talking terrestrially about myriapods, so centipedes and millipedes. And I see no responses to my questions. Can you hear me? Are you bored of coloring? Hello? Is anyone out there? Yes, you are bored of coloring. Yes, we can hear you. All right. If you want to throw in the chat, thank you for being able to hear me. If you're bored of coloring, if there's other things that you're doing in some other classes that have been interesting to you in these kind of pre-lecture moments, let me know. I can figure some things out. The word searches, I can make those. I can fill those in. And no, not bored of coloring. All right, where's Waldo? Yeah, I was looking for those kinds of things, and I suppose I could find like complicated biological pictures. And all right, that'll be my job for Wednesday. Note to self: make a Waldo finding invertebrate day. Okay. I will do my best to remember that for Wednesday. So, here we go. Let me unannotate that, clear all those drawings. This is what it looked like. These things that you've been coloring, these are all from a kind of a famous text of life, von Haeckel made in, or Haeckel made in the late 1700s, early 1800s, and these are giant, gorgeous lithographs. I posted the link like way earlier in the course, but it's a freely available PDF. So if you ever have like, I don't know, new rooms that you want to cover the walls of, these are, the Biodiversity Heritage Library makes this big text, a high-resolution scan of this big text freely available. So you could conceivably, if copepods are your thing, you could take the PDF and have one of them printed, because it's certainly out of copyright. It's been out of copyright for hundreds of years. And you could annotate your wall with giant lithographs of invertebrate life. Right? Because that's what you want to do. Now, thinking about invertebrate life is something that I imagine, like, when I imagine you, I imagine that you're thinking about invertebrate life all the time. And so when we were thinking about, let me see if I have just done myself in. I'm going to escape out of that. When we're thinking about invertebrate life, as I imagine that you have been, you're existing in a world that continues to change. And so lots of you have probably seen memes emerge out of the Evergreen, or the ship that's not called Evergreen, but that is currently blocking the Suez Canal and adjusting transportation of goods around the world. And I liked a lot of those memes, and I thought of you all when I made this morning. So this is sometimes, I think, how you might be feeling about this class and the amount of material in it when you think about the invertebrates, that is the ship and you are the excavator. And I think the thing to remember is I feel like that too. And why is that important? Well, it's important because there's no way that we can quiz you about the entire ship for the Ever Given. We're going to be talking about small, succinct bits, and we're going to be tests of the narratives that Sherry and I have kind of woven throughout the year, and those are what you'll be tested on. So keep going, little excavators. You will be successful. There's another, an odd computer scientist who has made this online. If you haven't explored it, it lets you move the evergreen anywhere you want in the world in a Google Maps. So this is actually, for those of you who remember what our campus looks like, this is what the evergreen would look like if it was placed on Johnson Green. So it stretches, it's a big friggin' ship. It stretches from the intersection of Gordon and College all the way almost to the UC. So this is a great big thing. There we go. That's the last bit about the evergreen that I will be talking about today. So, myriapods and crustaceans. We are, of course, in the arthropods when we're talking about this. And this is the view from Gerbet and Edgecombe. These are the two groups we're going to be talking about. So within this, we're going to be talking principally within the myriapods. We're going to talk about the chylopods and the diplopods. You know them as the millipedes and the centipedes, and you've known about them forever. And I think that's kind of exciting sometimes, that this is a group, these are groups, some of the groups today that we're going to be talking about, that you've known about since you were in kindergarten or in the early elementary school grades, and drawing things, and drawing millipats or millipedes and thousands of legs or hundreds of legs, and rolling logs and finding these flattened or rounded things. So these are groups, some of them in the myriapods that you're familiar with. So that's what we're going to talk about. The crustaceans, you're also familiar with. You know that they're delicious. We're going to talk about some of the ways that I find them surprising. And I'm going to acknowledge right off the start my terrestrial bias, because I'm going to talk to you about some terrestrial crustaceans in and amongst, even though the vast morass of this diversity, crustacean diversity, is in the ocean. But I'm going to talk about them because talking about myriapods makes me think about rolling logs and smelling spring soils in around this area, or in rainforests and cloud forests around the world. And when I roll those logs or turn over those stones, I find millipedes and centipedes and isopods, terrestrial crustaceans. So we're going to talk about those a bunch as well. We're going to talk about them phylogenetically, of course, because one of the things you saw, and I've mentioned it, but you see Crustacea there has an asterisk beside it. I've referred to these as kind of Tribbiani clades before, because they are in fact not monophyletic. So this is a word that we use, but we use it like this because phylogenetically it doesn't capture anything. The word that we're going to capture phylogenetically, kind of giving a spoiler away, is the tetraconata. And the only way that the tetraconata is a monophyletic group, or the only way that you could make crustaceans monophyletic, is to include all the insects and the hexapods. So, ta-da, we're going to talk about that today too. Continues to let people into the room, jumps back to the chat. All right, and we continue. So, with the myriapods, the centipedes and the millipedes are the groups that we're going to talk about, that we have some degree of specialization, but not limited compared to some of the others. So that there's two tagmata, two specialized regions, heads, and then a multi-segmented trunk, where the segments are pretty much identical. All of the appendages are articulate and uniramous. Uniramous is, we'll talk about that and contrast it with biramus. So you've seen this in the lab talking about crustaceans. They don't have a carapace. They have trachea and spiracles, and we think that those spiracles involved in respiration. We think that's things that have involved convergently with those in the hexapods, so they look similar, but it's really just about independent colonizations of the terrestrial environment. They have Malpighian tubules, again, probably also convergently evolved. Compound eyes, most with a lateral eye, some of them can't see at all. A very simple gut without digestive ceca, and they're males and females with direct development. This is from chapter 23. There's 2 chapters in Brusca that we're going to be going through today. So remember, when you're kind of touching up about these kinds of things, and remember how much time we spent in these 2 chapters, and spend kind of an appropriate amount, an analogous amount of time there. So here's my kind of summing up of that. They're compound eyes, but they lack crystalline cones. They've lost the median ocelli. They only have 1 pair of antennae. In the top looping video of the millipede, you can see that one kind of feeling its way around. And they have this Timishvari, which is a neat word for your Scrabble quiver. Organs on the head that are probably hygroreceptive, humidity, chemoreceptive, but not entirely known. We're going to start talking about the centipedes. And these are centipedes that you could probably go and find in your house right now. So this is a Scuttigera species that from Europe that moves around very, very happily within houses of North America. So moist environment. So if you've got a basement or in your washroom at night, if you've seen these very fast moving things across the floor, that's a centipede. You should be kind of a little bit pleased it's there, because if it's there and it's living, there must be enough other things there for it to eat. And that's one of the things about centipedes, is that they are predatory. Fast predators. None actually have 100 legs, which is, again, kind of a neat fact that you can wow your friends with out at the bars in the after times once we're all vaccinated. They have poisonous fangs or forciples that are up here. These are modified appendages. Predaceous and then flattened. When you see them, this is kind of a comparative thing, but if you roll a log, if you go out on Tuesday, I think it's supposed to be 16 degrees, unlike today, you roll a log and there's some slow moving but active things under there. If they look rounded like a loaf of bread, that's a millipede. If they look flattened like we'll call it a flatbread, that's a centipede. And the centipedes are the predatory ones. So there's the maxilliped, the appendage that is modified as the poisonous claw, that is about subduing prey so that it can consume it. Now, the scale of these, when you're thinking about these antenna or bearing terrestrial two-tagmata possessing animals, you're often thinking like this. They can be, of course, much, much larger.
But even whip spiders were not the most formidable hunters in these forests.
And just in case we're asked to remove this by the YouTube, we're going to watch a video, and I encourage you to go and watch it later on, but this is a video from Life in the Undergrowth, and we're going to get at least part of the way through cave centipedes. I love that. So if you love centipedes as well, and you love thinking about centipedes, then this is probably one to, again, tell your friends about. This one that's a cubit long, big as your forearm, and feeds on bats by suspending itself into their flight path in their caves. So, a little bit of the detail from chapter 23, some of the morphological detail. You can see these looping Malpighian tubules. Again, this is about concentrating waste and reclaiming water in advance of that. And even though it looks very, very similar to what we kind of sketched out in general for the panarthropods, the hypothesis, the current phylogenetic hypothesis is that this is convergently evolved. These Malpighian tubules look as amazingly similar as this, but they've evolved convergently in response to the tight evolutionary selective pressure of moving from a marine environment to a terrestrial environment. Now, associated with that is the spiracles. Now, the spiracles, which open or close the trachea on the hexapods, the insects, we saw even some in the spiders and chelicerates, like the scorpions, other euchelicerates on Friday. The spiracles here aren't super efficient because they can't be closed. And so that's one of the reasons that these animals, the myriapods, are kind of obligatorily restricted to moist environments because they're always losing water. They're also long-lived. So if you have a positive or negative relationship under a particular log with a centipede and you go back over the course of your undergraduate degree, you might be revisiting the same 6-year-old centipede through time. So there's the Malpighian tubules. A word, since we're talking about crustaceans, and I've mentioned eating some of the things that we've studied today, this is not a good group to eat. There's a bunch of different parasites that live within centipedes that can affect us very, very negatively. So uncooked specimens can contain a parasite, a rat lungworm, that can infect you and make you very, very unhappy. So there's another species as well, this nematode, Angiostrongylus cantonensis, that can lead to cephalitis or meningocephalitis in humans. So not a good idea to eat cephalopods. We could keep talking about these. Well, I could. So that's a good question. Why are they called centipedes if they don't actually have 100 legs? The same question you could ask in a second about the millipedes, because they also don't have 1,000 legs. There's none that have exactly either set. And so it's I suppose, a way of thinking about the fact that I sometimes characterize ecologists as counting 0, so none, some, many, too many to count. That's kind of the 4 numbers that an ecologist needs to know and needs to be able to enumerate in the environment. Taxonomists often count things with more detail, but I think what you're seeing here is that in deep kind of Linnaean time, 250, 300 years ago, what we were really doing was kind of splitting up groups according to, well, do they have 2 legs, 4 legs, 6 legs, 8 legs, 100, many, or many, many more, 1,000. And so it's a remnant of that. And I am looking at the Chat, so I won't verify anymore. And we're going to talk about now these loaf of bread shaped, the millipedes. I love this millipede and I love this particular view of it. You've seen it before, but this soil makes me think of the things that I can smell and how happy I am when I am there. But this is in cloud forests in northwestern Costa Rica. And so you're looking at kind of that wave, almost a metacoronal wave of legs, kind of that coordinated movement up all of those many, many leg segments. Now, see, these are the diplopods. So diplopods are called so because these apparent segments are in fact derived from the fusion of 2 segments. And that's why when you look at the legs as they move, the one on the video now on the bottom is actually in your dairy bush. This is another parajulid millipede. So each diplosegment, because it's derived from 2 independent segments, it gives the appearance of having 2 legs per segment. And 2 spiracles, which you couldn't see, but they're in there as well. And that's a remnant of developmentally the fact that these are, every segment that you see is actually a combination of 2 separate segments. So these are nocturnal detritivores. Their jaw parts are not very strong at all. So they're not good at defending themselves. They're not predators. They're not fast moving. Nocturnal helps as well in terms of the loss of water. It can. They tend to roll up, as you've seen, probably circles of them kind of spiraled up and protecting themselves. And so their protection from predators is really all about avoiding, to the most part, avoiding notice and making themselves noxious or not palatable. So they have a number of different chemicals. They're a veritable, called a kind of Walter Breaking Bad of the myriapods. They have lots and lots of different chemicals that they make, including hydrogen cyanide in one particular lineage. But these anti-prey chemicals include methylclone, or a hypnotic sedative that humans can take or have tended to take in the past. But these ozidines, which you can see kind of 2 little islets on that cross section through a diplosegment here, they can produce things like hydroquinine, quinine, different, where are we over here, benzol, different, all sorts of things that can be noxious or actually even burn. Certainly small organisms, but even large ones as well, like us. So they're kind of the gentle, slow-moving myriapod of that underlog community, but certainly not one that you'd like to pop in your mouth, I'm going to suggest. In the other cross-section, let me jump back for a second. You can see here, so this I pointed out here, the ozidines that protect or collect and produce many of these noxious toxic segments, but you can see associated with each diplosegment the gland that opens up and releases them from the side. And generally they're not leaking them all the time, they need to be handled. So you need to disturb them or tell it that you are a predator. Now, the majority of the ones that we've talked about so far, the quinine and the benzol derivatives, those are called type 1 and 2 glands. And that's kind of another view of them here from this Shear's review paper of myriapods. But there's another type in a particular lineage of millipede, a type 3 gland, which is analogous to something that we've seen, but not homologous. So convergently evolved, safe production of hydrogen cyanide. And we saw that in one of the mite lineages that we talked about on Friday. So you're looking at a dissected, so instead of the drawings here for the type 1 and 2 glands, take a look at this one up top here. This is a type 3 gland that has a reservoir, a valve, and a vestibule. And different chemical products, precursors to hydrogen cyanide, are produced on either side of that valve. So the reservoir produces the stable precursor mandelinitrile, and then when the valve opens, it's mixed with hydroxynitrile lyase, which the combination of those two produces hydrogen cyanide. But when they're maintained separately in those two different reservoirs, two different glands, they have no negative effect at all on the millipede. And a 1 gram millipede, which is relatively large, can actually produce 600 milligrams of hydrogen cyanide of a polydesmid millipede. And that's a lot because you only need about 160 milligrams to kill a small to medium-sized person. So there's a neat way to look at some of these cyanide-producing millipedes because they don't want, this is a relatively costly thing to produce, they don't want to waste what they're producing. And the ones that produce cyanide tend to be colorful. And I've kind of written a poll question here, a practice final multiple choice question about some of Appalachian millipedes that we're going to take a look at right now. So you've got two different things here to look at in this poll as I try and set it up in the background. The photo array on the left, there's two species of millipede that live sympatrically. Both species are you can find them by name in that adjacent phylogeny. They both produce toxic hydrogen cyanide. So what I'd like you to do is consider these statements and tell me which one of them is false. And we'll take a minute or so to do that. While we're doing that, I'll jump over to the chat, see if there's any questions. Not yet. All right, we're almost up to 50% of you. Let's get up to 80%. We're almost there. 38 of 50. Come on, 12 people. Let's go. Let's go. You got this. Evergreen ship. You understanding the invertebrates. Dig away. You've got it. All right, let's end the polling for a second. Well, forever. And share the results. Talk about this for a second. So, first of all, the majority of you thought the false statement here was that this is an example of Batesian mimicry. So, put your hand up in the air, pat yourself on the back. That is an example. This is the false statement. Batesian mimicry, remember, is an example where there is an honest signal about unpalatability in one species, and then in a different species, there is a dishonest signal of unpalatability that mimics that honest signal. So Müllerian mimicry is based around the idea that everybody is toxic. Everybody's giving honest signals. But then, and the advantage collectively when they're found sympatrically is that any one of them, the signal might be too dilute. The signal to noise ratio might be so biased towards noise that it would be hard over time for any potential predator to learn about the honesty of that signal. And so the Müllerian mimic, or the hypotheses of a Müllerian mimicry, is based around the idea that if more of you adapt the same signal, then you're all going to benefit because there's more teachable moments for predators who will be handling these hydrogen cyanide producing little colorful bombs. So it is a true statement that this is an example of Müllerian mimicry. It's also a true statement that for that to work, you don't actually need to see. And this is one case where these polydesmid millipedes have next to no eyes at all. They have photoreception, they can see light and dark, but they're not, this is not a sexually selected trait or anything where there are examples of where they're seeing these things. They both produce hydrogen, they're both toxic, and so this is an example of aposematism. So they are aposematic. So the bright color is associated with the honest signal. It's an honest signal of toxicity. So well done you. That's good. And so there's a question, where did you learn about mimicry? Yes, you've learned about it probably at a number of places in your biological training. We talked about it in 1070 at a number of points, and probably even earlier than that, perhaps in your high school career or in other classes, maybe in ecology, evolution. You would have talked about it for sure. So, but we go onwards. And you can go back and look at this. This is one of the links that'll be in the afterwards. But one of the neat things about this study that was published, that was derived from a study in PNAS, and this is a fairly small area in the Appalachian Mountains where there are many, many different species, independent, unique independent evolutionary lineages that all look basically like this, honestly signaling that they taste really, really bad. So before we move on to the crustaceans for the last bit, I have a couple more things I want to talk about with the millipedes. And one of them is the fact that there's actually not that many species described compared to some of the arthropod lineages, but there's many, many things that we have yet to learn. These are what were called dragon millipedes with these large spikes, so adaptations of each diplo segment that not just crypsis, so cryptic coloring, repugnatory glands, and also now these large spikes that try and prevent you from eating them. There's a look at some of the front end. So one of the morphological features that millipede biologists will talk about is this nanthocalarium, which is a fused appendages on the head and other mouth parts that is absolutely chock-a-block full. This is the Timashvari area, so full of chemosensory and tactile receptors. So 10,000 described species, but there's many, many, many more of them. Now, in Canada, a lot of the diversity that we actually have of millipedes, here's one from the dairy bush demonstrating this rolling defensive posture. Nearly half of the species that we see in eastern Canada, in Ontario in particular, are introduced. So the vast diversity is likely to be found in the tropics. Here's one example of near tropical, not actually tropical, but off in Tasmania. This is a species that was described a few years ago in Zookeys, which you're familiar with from your species naming assignment, where the species is known from an area of less than 12 square kilometers, and the type specimen was found in city parks in Launceston, so one small city in Tasmania. So much still to discover. One of the other neat kind of things about millipedes, and in particular with some of these, are another type of polydesmid. You can see these plates that extend over top of the legs and the coxa. That's one of the ways of kind of quickly looking at whether or not you're looking at a polydesmid millipede. These are ones that glow in the dark that we find in California, and it seems to be a really neat example that you can go and explore of where bioluminescence has evolved in this lineage in some California mountains. Initially, as a capacity, it appears that the molecules that are bioluminescing seem to be there passively. They've kind of hitchhiked along and become mixed up to no advantage potentially of the millipede. But their precursor molecules were ones that were involved in coping with metabolic stress associated with living in a hot, dry environment. And so they've been repurposed in evolutionary time in this lineage with all the species in this monophyletic lineage as a warning signal. So another way that a polydesmid has given itself a warning signal. And this is another PNAS paper that you can go, if you're interested in topics to write about later on in your biological career, that's where all these links come in handy. And then one last view. This is a rainforest, an Amazonian rainforest in southern French Guyana that I was collecting animals within nearly a decade ago. But you can take a look in that particular photograph at a young polydesmid millipede and its poop on the trunk of that buttress of that tree. You can explore it and find other animals like this hexapod, this braconid parasitoid under that spiny plant. And if you're interested in continuing to explore and finding more and more millipedes, this is a gigapan from our Algonquin Park. It's in our hallway, actually, in the science complex in the after times when we can go back there. You can go back and explore this one. Oh my. Let's see. Find this colorful polydesmid millipede in the leaf litter. This is in kind of northern Algonquin Park along the shores or closer to the shores of the Ottawa River near Brent Station. If you've had the opportunity, if you're from around that area or maybe have driven in from North Bay or just explored around. But millipedes, they share the place with us. They're amazing. And now I'm going to quickly confirm there's nothing else in the chat. We're going to move on to crustaceans for the last 20 minutes. Crustaceans in 20 minutes is kind of a challenge. They're a course in themselves. But remember, phylogenetically, I think one of the important things to take away from this is that this is a word, a biological word, that if in a future where monophyly means things to us because it lets us make predictive statements about biology because of shared inheritance, then crustacea is not a word that we should be kind of throwing around and using because phylogenetically it is clearly not a monophyletic group. You can see from the ostracods to the cephalocarids with the rhombidids, there's relationships that we're pretty confident within these groups, but the only way that we can group them monophyletically is to include all of the hexapods via this tetraconata. And so one of the interesting things to think about is whether or not you would consider, sometimes people have talked about crustaceans as being the insects of the sea. I think maybe a more useful way to consider it is that insects are actually the crustaceans of the land. And we can talk about that group because tetraconata or pancrustacea is this group where there is here, this node on the tree, shared presence of 4 crystalline cone cells in the compound ommatidia of both groups. So the apomorphy for the tetraconata, ommatidia in either the crustaceans or the hexapods, 4 crystalline cone cells and 8 retinal cells. So crustacea, there's a terrestrial isopod and some other marine ones. Diverse, more than 70,000, certainly more than 70,000 species around the world, also known to much of the world as food. And these are some of the characteristics, even though it's not a phylum or a subphylum, but as a group, these are some of the characteristics of the group. And they have all to do with about the lots of different leg characteristics. You saw this already in lab with Sherry. So we have a body composed of an 8-segmented head. We've shown you some pictures of this already in the last couple of lectures when we talked about the 3-partitioned brain and how different lineages of arthropods have different proportions or numbers of segments contributing to different roles, whether they're the chelicerae, the chelopads of the spiders or the euchelicerates versus the roles and the multiple sets of antennae that many crustaceans bear here. So there's a whole bunch of multi-articulate uniramus and biramus appendages that you see within. They have mandibles that function for biting, piercing, and chewing, gas exchange across specialized brachial surfaces, gill-like structures, or cephalized or specialized regions of the body. They have excretion via true nephridial structures with antennal glands and maxillary glands. They have a gut, a diverse and high surface area gut with lots of cecae. And they have a nucleus larva, which is not known in any of the arthropod lineages, major arthropod lineages. So this is kind of my summary of a lot of that. So they have a head and trunk tagmata. The trunk appendages tend to be biramus. Trunk segments have appendages. Suspension feeders use myxopods and a ventral feeding groove. They have a compound eye, 2 pairs of antennae, 2 pairs of maxilla. Let's talk about some of these groups now. So why do we call them food? Well, simply because most of the world's marine systems are obligately dependent upon crustaceans and the vast volume of crustaceans. That colorful set of, that lithograph of species at the very first slide was we listened to I the Mountain sing The Boat as we arrived in lecture today. Those are copepods. And it's presumed, there's been one kind of back of the napkin calculation about the density of copepods in the world that's calculated 1.3 times 10 to the 21st power number of copepods living in the oceans. And that's super conservative because that's assuming 1 copepod per liter. And there are many orders of magnitude more than that. So you could take that to moles and moles and moles of copepods. They have some of the largest and some of the smallest examples living in the oceans. They can live terrestrially, marine, they can be freshwater, they can be parasitic. And we're going to listen to Attenborough tell us a little bit about them here. Smallest, this is we've talked about the Small World photo competition before. This is a copepod less than 1 millimeter long. That was one of the winners a bunch of years ago. But they can also be amongst the largest arthropods or invertebrates. This is a decapod that can be up to 4 meters long and weigh 44 pounds, the Japanese spider crab. So the groups, the lineages within that we don't have time, that warrant a class of their own, and potentially that you can dive into further if you take limnology, and if you take the Huntsman field course, you'll have time to think about different lineages of crustaceans. But the ostracods, so these are basically little more than a head, 5 appendages on the head. These are tiny, tiny ostracods. The branchiurians and the pentastomids, these are ectoparasites that you might think about and have to deal with if you get into aquaculture, freshwater and marine fishes. The brachiopods, you probably know, you might have seen them. These are like Daphnia, so there's lots of freshwater examples of them. The thecostracans, these are barnacle-like crustaceans. We'll talk a little bit about them. The malacostracans, including decapods, so these are ones that you're familiar with probably in a lot of your menus. So lobster, shrimp, woodlice, so isopods are in there. Copepods, 1,200 species, marine, freshwater, parasitic. These are really the dominant group that make up the zooplankton of the world. The Remipidia, oh, this is a super not diverse group, only 10 species known from freshwater caves, and we think they morphologically look like what an ancestral crustacean. They bear a lot of have retained a lot of ancestral traits. And the cephalocardia, these are epibenthic deposit feeders that have undifferentiated trunk appendages. So in as much time as we have, I'm going to jump into some of these. Talking about the legs, for example. So one of the things is that we have suspension feeders that use myxopods and a ventral feeding groove here. So we've got exopods that are endopods. So the combination of respiratory surface, a walking leg, a little bit, and creating a feeding current so that the food particles are moved up. You can see in the bottom image. So the so the stenopod there used is for walking. The exopod is the phylopod, and the phylopod is like for currents and for creating currents and for swimming, and then moved up into the mouth that's meeting these tiny little particles. Now Daphnia, that Branchiopoda group, this is in aquatic systems, like if you're in and around the Guelph area and you go out in the next few warm days into the Arboretum, into the Goose Creek Walk and lay on the boardwalk out there, you'll probably run into some zooplankton. So they're a critical part of the food webs of lakes and ponds, and they can occur in great, enormous densities. And I mentioned I mentioned earlier taking the Huntsman, the marine field course. You can also run into to crustaceans in both Algonquin Park, like this. So taking field ecology locally or more locally in Algonquin Park when we can do that again together. Or even going up to Churchill, where there's actually been quite a bit of work done on Daphnia that live in pools that are in the rocks that are adjacent to Hudson Bay, which is where I filmed these Daphnia teaching the Churchill field course a few years ago. And I mentioned field courses not to make you feel bad about the fact that they're not offered this year, but just to remind you to keep that part of your academic dream alive and with you, so that when we do offer them again, that you are there. So one of the things so we talked about the fact that we've got a cephalothorax and an abdomen. So two tagmata, basically head and a trunk. That's what I call the cephalothorax, and the abdomen there. And so the apomorphies for these, for arthropods in general, but in particular for the crustaceans, are really about differences in segmentation and about the function and role of the number of appendages. And we'll talk about that in a couple of different ways here. So first giving you some names about the head. So this is the acron, plus the first 5 segments that are fused together to make this capsule, the head and the cephalothorax. And then the trunk, this remainder of the body. So 5 pairs of appendages in the head, but then all of the remainder of them across the back end of the abdomen. So the head with 5 segments, 5 pairs of appendages, and they get broken up in terms of 2 pairs of antennae, mandibles, and then 2 pairs of feeding appendages, these maxilla. So there we've got 2 antennae and mandibles and maxilla. So food manipulation. Now, for the malacostracans, much of the diversity of crustaceans as we experience them as humans, we see kind of an interesting pattern, an evolutionary pattern across different lineages of an evolution from being principally pelagic to principally benthic. And cool story, hello Dr. Jacobs, of decapod evolution that goes essentially so pelagic to benthic, one from swimming to crawling. And the largest morphological change here that you can see is if we track it from shrimp, pelagic, crabs, benthic, is the abdomen, which becomes very, very shortened and tucked up under the cephalothorax. So the carapace gets expended back over it, and then buoyancy changes. So they go from neutral, so floating, to negative buoyancy, so they can walk. And we see the evolution of sideways crawling. Now, this is a diagram from the older text, Rupert, Fox, and Barnes, and I can colorize it here to make you kind of look at some of these similar areas. So here in green, we see the head appendages and the emphasis that changes as you move in evolution from being pelagic to terrestrial, and then the abdominal segments that get minimized and tucked right up under that. So maybe this is in the after times if you're going to go out to, if you live coastally, go to any of your local restaurants, and if you live terrestrially in the middle of the continent, go to Red Lobster or something, and order a bunch of these things, lay them out on the plate in front of you, wear a nice bib, cover them in butter, and maybe trace out this pelagic to benthic. That would be a great lab exam. That would actually be a really tasty lab exam. That's going to distract me now for the rest of the morning. So they're gonochoric, crustaceans are, with internal fertilization. And you can see the gonopod that many males use to directly or to transfer sperm to the seminal receptacle in females. And if you handled her in lab, I know you had a crayfish cam for a while, and if you flipped over those, or if those crayfish were flipped over for you, you can actually identify males and females. You can see these seminal receptacles in gonopods. And as you can imagine, the attractiveness and the signals that males and females send to each other differ if you're a terrestrial or a marine lineage. That the marine ones tend to use pheromones that drift along in the water, whereas the terrestrial ones tend to use visual and auditory signals. And I'm touching on that only briefly because entering into that can be some of the way that crustacea becomes a class on its own. Giant isopods. Isopods are we're going to spend, I think, maybe the rest of this lecture. Isopods can be marine. This is a study, actually, of decay where off of Florida. So this is what Florida people do, Floridians do when they're not making the news otherwise. But what we're looking at here is a already dead alligator that was tied onto, weighted, anchored, and then cameras put on it to look at who's eating what and decaying it. And this football-shaped thing here, this is an isopod. This is a pill bug. So we know these, you know these if you grew up in and around this area, because of their, they exist probably, again, if you've got those Scutagera centipedes in your basement, you might have isopods as well, and they might be getting active right now. They act where I work in Costa Rica. These are ants that are delightfully eating a lot of isopods in cloud forests. So crustaceans as food is not just a marine thing. In cloud forests around the tropics, crustaceans are also food. But the terrestrial, and so this is the isopod, this is the isopod here, the gray one. This is an amphipod, so another lineage that lives terrestrially. But for lineages that are pretty much obligatorily marine or freshwater, making the move to the terrestrial environment has had a lot of challenges. And so we tend to find them only, like I was mentioned in passing, these two videos are in cloud forest. So those are areas that are just wet all the time. So they are very close to being a freshwater environment. So their cuticle, how do they manage to live? And this is one of the challenges that they've surpassed, because they have limited wax on that epicuticle. Remember, we looked at a cross-section of arthropod cuticle. And that 3% of epicuticle, of waxy, it's not a huge part of the, proportionally, in the size of the cuticle for these arthropods, but it's very, very important because it's how you prevent a lot of water loss. And so isopods basically get around this by behavioral adaptation. So you see the EC here is the epicuticle. So limited, it's very, very small. So if it's 3% on average in that generalized diagram that we shared earlier, thinking about arthropod cuticle, it's even less within the isopods. So they have some other advantages. So their gills are exposed exvaginations in here at the base of these walking legs, and these would become dried out very, very easily. And so they're protected by what's called pseudotrachea. And so these pseudotrachea have spiracles, and then this kind of almost an amalgam of a gill and a trachea that can tolerate much drier conditions, much less humid conditions than can many of the more obligatory freshwater species. And so we see the gill there, and then a view from your text up at the top of the pseudotrachea kind of being bathed in the hemosyl with a spiracle. And so the pseudotrachea is an adaptation then, behavioral adaptation, so live in a humid place, and a pseudotrachea rather than a gill, are two adaptations for terrestrial isopods. And these, again, this seems to be the trachea as a convergent thing amongst the arthropods in general. They have another really interesting adaptation towards living terrestrially, and that is this area right here, which is called a marsupium. And this is a brood pouch where the female retains, you can see under these layers, and the layers there, those protective layers, are different parts of the appendage that have grown out to basically make like a little pouch, the root marsupium, where the eggs and then the first instar larva are reared. So they begin in water, in a very completely moist environment, and then they finish in air. So this is an example of extended parental care within some of those terrestrial isopods. So crustaceans we could go on and on and on, but the clock says that we can't. So that's our Monday. I hope that you get a chance to take advantage of some of the sun that's supposed to be coming to us on Tuesday before the freeze that's coming later on. And take care of yourself. Do something good and positive for your body and for your brain, and do things positive for the brains of the people that you're living with right now. And we'll rejoin each other on Wednesday. I'm going to be around in the chat for a second if there's questions that I've missed. So take care of each other.